Dr. Marianne Ellis is an associate professor in biochemical engineering and the head of the chemical engineering department at the University of Bath. We are super proud to have her as a research grantee through our competitive research grant program, and are really excited to hear about some of the very important research she's advancing in the realm of bioprocess development and bioreactor design, specifically for the low-cost, large-scale production of cultivated meat. She has a lot of experience within academia and with commercializing the technological findings from her research, specifically in the realms of tissue engineering and bioprocess design. And she has a lot of experience working within the cell therapies realm and with cultivated meat production. So we're really excited to have Dr. Marianne Ellis here with us today to share her expertise. As you all know, a lot of the cultivated meat technologies come or borrow from the biomedical field, but two unique challenges that are kind of unique to the cultivated meat field are the the low cost of the products we're trying to compete against, and the sheer scale and magnitude of the volumes we'll have to produce cultivated meat at in order to feed the world. So Dr. Marianne Ellis will speak about how her research helps us contemplate paths forward to overcome some of these challenges. And I'm really delighted to have her here with us today. So Marianne, whenever you're ready, please feel free to take it away.
Thank you, Amy, and hello, everybody. Just bear with me while I get the slides shared with you. Okay, Amy, is that good? You can nod, I can see you. You can see those
that
okay?
Looks great.
Yeah. Okay. Well, yeah, thank you to Amy and to GFI for inviting me to speak to you this evening or this morning or tonight, wherever you may be. So as the title says, I'm going to talk about designing large-scale bioreactors in particular and bioprocesses for cultivated meat. I would say at the outset, while I'm here giving this talk, this is very much a team effort. I've basically added the names of the team members and collaborators who have contributed to this work as we go through. So please do recognize this is very much a team effort. Just to give you an overview, Amy just done a brilliant introduction for me, so thank you, Amy, but I'm just going to say a little bit more about my career, which will hopefully help give you an idea of my perspective on the challenges that we're facing in our field. Given this is a scientific, I guess an expert audience, but I'm anticipating that not everybody has bioprocess design expertise and know-how. So I am going to do a bit of an overview of bioprocess design, just to kind of for those who are less familiar with it, and then talk about our conceptual bioprocess design, and then some of the detailed, some new, very new, in fact, results. So actually three or four papers that are actually written but not yet submitted. So hopefully it will generate some conversation with some of our new findings, and then of course with some conclusions. I've also we'll come through. We'll see in a minute. I'm also going to share with you some of my current thinking, not resolved thinking, but current thinking on some aspects I think we need to be thinking about when it comes to our field of process design, which is hence the brackets in here. Yes, it is about scaling, but that's not the only consideration when we're looking at scale up. So just to put a little bit more kind of context on the intro that Amy's just given you, I started my academic career as a lecturer in 2005, having done a PhD and a postdoc in tissue engineering for bone regeneration. and so designing a bioreactor for that. Done a number of, worked in a number of areas, basically using platform technology, bioreactors for cell therapies and non-animal in vitro models. Another area of my of significant interest for me is using organoids for drug discovery, and basically providing organoids for drug discovery. I'm an experimentalist by trade, and the majority of the work in my group is experimental work, so wet lab work, but we also do some computational fluid dynamics, and certainly I've worked pretty much my whole career with mathematicians and biologists and other natural scientists, as well as working with people in social science space as well. So that's since 2005. I really got interested in cultured meat, cultivated meat, since about 2013, looking at bioprocess design, but inevitably my background with bioreactors has led the focus of our research to be on bioreactors and scaffolds, because that is a combined part of the design. So what's my perspective on this? So my vision for working in this space, why am I doing it, is because I would like to be part and help with what I can to contribute to food security, so that essentially everyone everywhere can have a protein source, and cultivated meats and other cellular agricultural products, of course, can do that. But from my background, as you've just seen, really actually what my team and I are trying to do is solve the problem of how we can make affordable cultivated meat in a zero carbon process, a net zero carbon process. So just to, again, put a bit more context around this, and this is important for process design, and I'll say this a few times, essentially because you kind of need to know what you're aiming for to be able to do the design well. There's a number of things on here, but particularly considering the decarbonisation of the food chain, and that is important for scale up. It's about making something viable on a large scale, but we do need to be able to do this with the low carbon cost, and I think everybody would appreciate that, and also in a way that is culturally relevant. So when you're thinking about your bioprocess design, thinking about the market around it, where's the plant going to be built, who are the customers, and all of those things will come into your design choices. Again, I'm going to touch on this a few things, and this is something that you will all be familiar with if you're already working in this space, and the potential benefits that we are expecting from cultivated meat. But we collectively, as a field, I think we talk a lot about climate action, essentially, and somewhat about zero hunger. I'm going to put it to you as the audience that we also need to be considering the good health and well-being and the product formulation at the end. I don't work in product formulation, but as a bioprocess engineer, what we are designing it will be impacted by what people want to do with it. So considering the end product is important, and I believe it should be something that's healthy. So just to pull that together, the challenge that we are facing is the industrial scale production, but it's not just about size, although that is clearly very important consideration. When we're looking at scaling up, we also need to be looking at net zero carbon processes, looking at animal-free raw materials. And I would also say the healthier diets. And for me, this all comes around to the triple bottom line. So as a field, I think we all fully understand this. Probably most of us joined this, at least in part because of the planet aspect. there's certainly opportunity for profit. There's a lot of companies in this space now, but healthier diets relates to people. And that is part of the triple bottom line as well. And when we are thinking about how we design a process, I believe that should be taken into account too. So this is going to be the overview of process design, really aimed at those who haven't had any background or training in process design. So it's just going to be an overview for those who are interested, and help with some of my other slides. So we're using tissue engineering techniques, which is essentially mammalian cell culture techniques scaled. When you're working on a lab scale, you've got lots of people doing very manual jobs. It leads to batch variation. So you don't have the consistency, and it's not efficiently scalable in terms of cost or quality. And that is why we have bioprocessing, because it automates, or certainly semi-automates, the process. By having the process control and the reduced human intervention, you end up with higher quality products. The design of the process itself enables you to reach those higher volumes. And you ultimately get the same amount of product for the same manual input, which, for those of you who are already at the stage of looking at the manufacturing cost of this, people are an expensive resource. So if you can reduce the need for human intervention, that's going to help with your costs. And this figure on the left-hand side here really is from a book from Wheelwright, fairly early, haven't got the year here, I think it's 80s, bioprocessing book. And you're looking at an ideal process that combines low cost, high quality, and fast to market. So that's your optimal process. Certainly these here will come down to process design. This is more of the business focused aspect. Okay, right, yeah. So just to touch on the considerations that need to be accounted for in process design, we need to understand the cell biology. We are looking to, in the first instance, replicate the in vivo environment, and potentially, and almost definitely, we will need to adapt cells to the culture environment. If we look historically at CHO cells, other foodstuffs like corn, there's been a development of the cells that are used to enable an efficient process for products to become, or those cells to be suitable to become industrially viable. I'm not going to go into the biology here. Really, this is just to highlight that there is some complex biology behind muscle cell growth, both proliferation, differentiation, and then hypertrophy and the muscle deposition. And it's important that we understand that, particularly if we want to adapt cells to the culture environment. So the next couple of slides also on just how to do process design. It's essentially for anyone who's here thinking about, I don't know if anyone kind of pre-degree thinking about what they want to do for their degree in the following years. This is a sort of thing that you would do as a student, undergraduate student, on your first degree as a chemical engineer. You start here with the final product spec, which is why I've mentioned already the importance of knowing what that final product is, and in my personal opinion is one that is a healthy final product, because that then helps you work out what you're creating or you need to produce, and then you can work forwards. You need to understand the biological data. You do some calculations for the bioreactor, and then you essentially go through this process, work out how much media you need. This box here is actually iterative. So you're selecting the bioreactor, sizing it, sizing the equipment around the bioreactor based on the essentially the inputs and outputs of your bioreactor. So this is things like your downstream processing, your media storage tanks and recycling. You also need to understand how you're going to start up and shut down your procedures, whether you've got batch, semi-batch or continuous. And then you've got your hazardous operation studies. So it's a very important part of process design, making sure that your plant will be safe. The environmental impact, and then everything around that would be your logistics, infrastructure, personnel requirements. From all of that, you'll get a cost, and it's likely you'll go around this cycle a few times until you can build an efficient process. So just to give you a visual on the considerations for designing the bioreactor alone, these are some considerations that Scott Allen, one of my PhD students, has pulled together. So in the grey boxes, you've got the reactor type. There's a number of different types that you can choose from. You then need to take into account the energy, inputs and outputs, the material balance, so the materials that are going in, waste products and byproducts that are coming out, and the cell environment itself, which links to the scaffold. All sorts of things to take into account, including cleaning and downstream processing. So just to say, really, this is to give you a visual of there is actually quite a lot of complexity in designing reactors, but actually, as Amy said, there's a lot we can learn from other industries, which is very good news for us. So on to the conceptual bioprocess design. This is an example that if any of you have heard any of my talks before, then you might recognize this. We have updated versions, but the principles are the same. If we just move forwards to show you what all of these various parts are. So we've got the bioreactors here. But when we're looking at that kind of flow diagram of how we go around designing it, so you've got the bioreactors here. You've got waste streams, potential for waste valorization, so creating more valuable products from the waste, here really from lactates, and looking at recycling, particularly water. You can recycle. You've got your media formulation. There are decisions to make about that, but then there's also when you've got the recycling and the new components coming in, and you've got filtration requirements or sterilization requirements. There may be potential for combined heat and power. Depending on the external environment that your factory is built in, and the various temperatures and heat flows that you have in your system, that you might be able to essentially save carbon and cost by having combined heat and power. Just to give you an idea, if this plant was built in Wales, which is where my colleague's farm is, or Iltyd Dunsford's farm is, then we have a balmy annual temperature of 11 degrees. But if we're looking at Turkana, so there's a refugee camp in Turkana, Kenya, and there the average temperature is 30 degrees C. So you can see you end up with very different energy balance requirements, and then knock-ons to this the combined heat and power. So around that, you also have, as I've mentioned, the design optimization, infrastructure, life cycle analysis. I will touch on that briefly in a sec. And then the social science aspects of that around consumer acceptance and public engagement as well. So this is a simplified version of that, and this actually is relevant in a couple of slides' time, because this is what we have. We've got a new LCA paper coming out, and this is the version that you will see in there. But if we pull all of that down into something that is more manageable, so we essentially have the bioreactors here. So I'm a bioreactor like researcher, so I would put the reactor at the center of the process. I have colleagues who are downstream process engineers and who would put the separations at the center. But ultimately, this will just give you a simplified view of what we're considering, including the recycle stream here. And say the heat exchanger for going into the reactors here. And the final product formulation. So I'm going to just talk very briefly really about this, but just for the to look at the whole picture of the process design before getting on to our recent findings from the lab. So taking you through, I guess, the years and the progress of work that Hannah Tuomisto has been involved in, and been working with Hannah since, in fact, it was Hannah who introduced me to the whole field of cultivated meat. So Hannah's very early work, and we know, and she knows, that there are assumptions in here that are not valid, and so there was an update. So after we first met, we did an update in 2014 for a conference paper to that, and you can see here that so this is for Wales. So you can see here that on the whole, cultivated meat is favourable, except for the energy considerations. So this early work allowed us to focus our lab work on addressing this question. This also was done, so 2014, so the process I've just showed you was 2017. This was also without recycle, and so with recycle you expect to see this lower. So again, we all know this, but there is great value in working with people who do modelling, so we can help to identify where to reduce our carbon requirements and the like. So on to the updated, or a very small summary of our updated work that we're hoping, well, our goal actually was this Friday for submission. We'll see if we make that or not, but essentially what you're looking at here is one example of the output. So through Scott's work in the lab, we've got some basic data for C2C12s in culture plates, and we've combined that actually there's a whole very large number of scenarios. So this is really a summary of some of those. So this particular graph is showing the climate impact compared to the baseline conditions, which you can see here is for C2C12 cells using well plate metabolic data. Okay, cultivated in hollow fiber bioreactors using DMEM with FBS. This is a base case. 16 days proliferation, 7 days differentiation, and 0 mass growth during differentiation. So what you can see here is a summary of essentially the differences to that base case with all these different considerations down here. So I'm not going to go into these in detail, but basically you can see that, firstly, there's quite a lot of variation, but ultimately what we have seen, and this is not going to be surprising for this audience, is that media is the biggest contributor to climate impact, and amino acids are the biggest contributor to media. I think there's essentially all the studies now are pointed in this direction, just to highlight that recent work. Okay, so now moving on to the reactor design. So just pulling out from that large process design that I just showed you. So bioreactors designed well will allow us to use less media, less energy, less labor, and less cost. We also need to take into account the media formulation and the raw material sourcing when we're really looking and considering about net zero carbon. And then from tissue engineering perspective, bioreactors design is very closely linked to the scaffold design, hence the two underlined there, and the cell source as well. So primary cells versus something like C2C12s, which are very culture adapted, and potential new cell lines as well that are generated specifically for our field. So you can see this, you end up with a interlink between all of these, but I'm just going to focus today on the scaffold and the bioreactor in particular. And I said, we're having to work with a number of people to develop these various aspects, but ultimately, we're aiming for high protein density, so something that is scalable, affordable, and it's the protein really that we're interested in. So again, this is more for those who are less familiar with process design and bioreactor design, just to give an overview of the different kind of options that are available. Cell culture flasks are nice and easy to use on a lab scale, but they're really not a very efficient system. They take up a lot of space, and their relative volume is actually very large. Now, there's all sorts of different systems. So stirred tanks are common and standard in most biochemical engineering industries. Fluidized bed bioreactors have an improved volumetric relative volumetric capacity compared to stirred tanks, and hollow fibers even more so. Essentially, hollow fibers and fluidized beds are more complex to design and operate than stirred tanks, and part of the process design is a balance between complexity and actually the scales that you want to go to. But ultimately, when you've got a higher cell density, you will get both CapEx and OpEx savings, floor space and energy requirements savings as well. Floor space is, so footprint, land use is important, and particularly if you're looking to build a plant in an expensive location. So for example, we've just finished our recent process design with our master's students at Bath, and this year, off the back of our links with Singapore, and also just very exciting news recently, we did the design this year for Singapore, and the cost of land in Singapore is extremely high. So being able to have a compact process is really important, and you would say the same if you're thinking about a process that's going onto a space shuttle, for example. So Amy mentioned this, I mentioned it earlier. This idea of repurposing is actually very important, and this is an example of some of the modeling work that we've done. It was actually for a liver bioreactor, but the correlation here of the pressure, flow rate, the feed to permeate rate of the hollow fiber bioreactor, and basically these are the properties of the fibers, is actually still relevant. So we don't have to start from scratch, which is really good news. We can repurpose what is already out there and innovate on top of this ourselves. But these types of correlations allow us to correlate the biological and physical data that we have. So in this case, looking at the oxygen profile along a hollow fiber bioreactor, to allow us to set the operating conditions, so the flow rates using back pressure to set the transmembrane pressure, and then we can ensure that all cells in this system will receive the minimum amount of oxygen that they need, and because of the way that this model is set up, it allows us to scale, because we can essentially calculate the gradients along the fibers. Okay, so a bit more on scaling of the bioreactor. So as I said before, it's not just about size, although that is, we do need to take that into account. So it's net zero carbon. Okay, so we use carbon within a process. It's net zero carbon. How can we ensure that we are not taking more from our planet? So that is the focus of this talk. Animal free is, so in terms of the media I'm talking here, it must work in the bioprocess. If we're looking at essentially serum free media, then that formulation needs to be stable in a bioprocess. It needs to be stable in dynamic flow, and it may need to be stable in storage conditions that are, maybe not what you might want to have in the lab. So, it's if you've got a lab, even a small scale facility, you can have a fridge, you can have a freezer, and it's quite easy to keep things cold. But is that still going to be possible if you are in other environments? And then as I mentioned, I touched on this, the healthier diets question. And this is kind of a question for us and for this audience, perhaps, is do we have, as a field, have a moral duty to drive change towards healthier diets and this concept of the triple bottom line? Because we could continue to design processes. Now, the burger and other similar kind of fast food items are well known, and it's been really important for us, to demonstrate that cultivated meat can be made and can be eaten. There's not the only things that are made, but that really is how I feel the world sees what we make. That's not necessarily what we might be aiming to do, but I think there's possibly a time now that we need to be considering the diets of people, so that we can contribute fully to that triple bottom line. That's actually all I'm going to say on that. As I said, the focus is going to be on the net zero carbon. So first of all, some of our recent work from Saeed Mazinani, who is our postdoc on our GFI project. So we, some of you might be surprised at this, but actually polystyrene is a good material to use in terms of net zero carbon, because it's actually relatively easily recyclable. Had a very interesting conversation with a chemist about this, because actually I'd actually worked with him making plant-based biodegradable fibers, and actually polystyrene, we for good or for bad, we have polystyrene out of the earth. We have it in our system, and it is reusable. I mean, the fibers don't degrade, so you can reuse the fibers themselves, but you can then basically redissolve them and reform them again. And actually, it's the polystyrene fibers that we use in the LCA study, that in the paper that Hannah will be submitting soon. So to make hollow fibers, these are what I would say standard size hollow fibers. They're 800 micron to millimeter in diameter-ish. This is a very nice example of the structure that you can see here. You can see that the porous structure. These particular fibers are microporous, made. So Saeed, who has great experience in hollow fiber membrane engineering from his previous experience before joining this project, has actually found a pretty novel combination of a solvent and water to give this microporous structure, which has improved permeability and permeance properties. So we've already used polystyrene, and you do need to oxygen plasma treat it. So there's a publication here from a couple of years ago now, again, where we're essentially repurposing this approach from what we did with our liver model. But polystyrene is basically tissue culture plastic is polystyrene, and so like the flasks you buy, we oxygen plasma treat it. So just some data on that, is that you can see here we've got some early data on the permeation. So we've got, this is on the permeant. So we've got, essentially this is the permeant of bovine serum albumin. Albumin being an important component of serum. It's essentially a carrier protein, and it is in abundance. But what you can see here is that there is a slight drop. This is only over 1 day, but you can see there's a slight decrease in membrane permeance, essentially the flux or the amount of flow across the membrane, across a certain area of a membrane in a certain amount of time. So why is this important? Well, when you're designing your process, if you just measure the properties of your membranes in pure water, and then you set your operating conditions around that, and then you add in protein solutions, and you're going to get pore blocking potentially, and reduction in supply of media to the cells on the outside of the hollow fiber membranes. So your raw material requirements, your cost, your pumping cost will not be what you would have predicted with your pure water systems. So those fibers that I've just showed you are these larger standard size fibers. They typically have a smooth surface, and when you grow cells on them, they will just be random. We've also got some work here with Patricia Esteban, who is a lecturer in Aston from her time in Bath, where we've made microfibers. So first of all, these are much smaller, so you get an increased surface area for each fiber. But the really lovely thing about these is they have these striations on them, which we see that C2, C12s align with when they proliferate and they grow. So Scott has put essentially some numbers on this. What you've got here are the cells that are control cells, so in a flask, and what you can see really is that there's no alignment. They're just, all these cells just kind of randomly spread. But with the grooved topography, you can see that actually you have very good alignment of the cells. You've got here 3 days and 6 days with the black. So on the whole, striated material is good. Okay, so it's a structure that we also see on grass, and again, various materials. I didn't see everybody who was popping up on the screen, but I did see Glenn was in the audience. So a shout out to Glenn, whose work on decellularized plants and scaffolds for tissue engineering is fantastic. And this was our kind of example of this using grass. So grass in the UK, most of the year is a waste material. We have a warm, wet summer, and we literally can't get rid of it fast enough. So it's it's sort of almost cultural sort of location relevance that grass could be a good scaffold for this field in places like the UK, where we just have a lot of it being grown as a waste product. But you still have to ensure that you're correlating your biological and physical parameters. And as you can see here, we've also get really nice alignment with the cells along the grooves of the grass. This isn't a particularly special grass, I'm afraid. I couldn't tell you the name of it. It's standard garden grass from out the front of our building in Bath. Okay, so that's kind of the scaffold aspect. I'm going to now move on to dynamic flow and media supply. So for those of you who are familiar with well plate culture and less familiar with bioreactor culture, then this is a good place to understand the beginnings of dynamic and bioreactor flow. So if you were to seed cells, and thanks to Scott for pulling this really nice slide together for today. If you took cells, you put them in, you seeded them in a plate, and you didn't change the media, you know that you get yellowing due to no media change, and you'll get some growth, but it's not going to be that good. Now, if you do a daily change, all sorts of regimes you can do, but if you change the media daily, yes, you do still get depletion, you do still get waste products, but you will get an improvement, an increase in the cell density. So we're looking here at cells per centimeter squared. And this is essentially a semi-continuous process. So you can get those higher cell densities, you're removing waste, you're replenishing oxygen and other nutrients. Okay, so let's now take the next level of consideration for media supply and culture conditions. It's the question, often come up, exactly the growth rate of cells in certain conditions, the glucose consumption rate, lactate production rate. So again, this is mainly Scott's work. So what you're looking at here are three graphs. Each of the orange lines is related to lactate, and the blue is related to ammonia. So the concentration on your y-axis is the starting concentration of either lactate or ammonia. So these are threshold experiments to look at how the cells respond to already having concentration of these waste products in the media, remembering that they will be producing these as well as they grow. So you've got the specific growth rate here. You've got the glucose, the specific glucose consumption rate, and the specific lactate production rate. What these graphs show collectively is that the higher ammonia concentrations result in the C2C12 cells growing more slowly, but also using more oxygen. Okay, whereas higher lactate, they grow more slowly, but they don't use more glucose. Okay. Also, interestingly, is that you get this steep drop between 2 and 5 millimolar of the starting concentration of ammonia in terms of growth rate, but it's actually more or less linear with lactate. Now, we're still bottoming this out, and we are still yet to give, I guess, a definitive stoichiometric balance, and the metabolomics and the metabolic behavior is complicated. But I think it's quite clear that the importance for optimizing your feeding strategies in your bioreactors, plus the recycle rates and designing the downstream processing, so separation units, this will link back to the how the cells respond to the media, and this also gives some indication to help us adapt cell lines to culture conditions. So okay, if what we don't want is this drop here, then can we select for a cell line that is less sensitive to ammonia, for example? Okay, so on to looking at some of our dissolved oxygen experimental data. So again, this is Scott's work. So he has taken some readings in the reactor with cells in. Let me just orientate you around here. So we've got 5-day culture. We've got dissolved oxygen on the y-axis, and then we've got these different kind of standard conditions. So starting at the top is the solid black line, is the bioreactor inlet dissolved oxygen. And just for interest, he's added here the dissolved oxygen in arterial blood, and also the pericellular dissolved oxygen, and these are taken from literature. So he ran the reactor in 3 flow regimes. So we've got the red line here is diffusion dominant, so meaning that the flow of oxygen from the lumen to the cells on the outside of the fibers is dominated by diffusion. So these other 2 cases are where we've got the blue line is advection and diffusion balanced. So we have the movement of oxygen molecules across the with pressure driven, and then we've got advection dominant. Okay, so very, very similar here actually. So the take-home message here is that by having the advection or advection diffusion balance regimes, then we are maintaining that that oxygen level far better than if we have just diffusion alone. So I mentioned that we do some CFD, some modelling in the group. So Saeed Mazinani has been leading on this and working with Scott. And this is really just an example to show how the model that they have been developing has been experimentally validated by the experimental work. So again, working in cells per centimetre squared, because that essentially you can scale that. You can see here that the dotted line is the model. The black solid line is experimental data, and just to show how that links to the cell growth, then you can see there's actually the inversion of those curves. So Saeed's also been looking at shear stress. So we know, so hollow fiber system where you've got flow through the lumen, you've got the membrane, and you've got the cells in the extra capillary space, protects cells from the shear stress of the feed. And but that doesn't mean there is no shear stress on the outside. And Saeed has done some modeling to look at this. You can easily, if at all, measure this spatially along a fiber, certainly not every single one. So what you can see, if we've got the inlet to the fiber here and the outlet of the fiber here with 3 different flow rates. So these are flow rates that that Scott uses, and we've got the mean lumen velocities there, because I know some of you will be interested in that. You can see that as you increase the flow rate, you can see, even though the shear stress is still very small, you can still detect that you'll have a higher shear stress at the on the cells closer to the inlet than at the outlet, which is not really surprising, because if you look at the pressure profiles in here, you're going to have a slightly higher transmembrane pressure drop across here, so this will result in these higher flow rates. So nothing actually in terms of design, it's okay. These are relatively low, but it's important to know and these are with our micro porous hollow fibers. If you increase the pore size significantly and you've got more advection and you've got more flow, you may get to a point where your flow is such that you're shearing cells off. So there's design work that can be done at this level. Ultimately, fibers, as I said, do protect cells from the direct effect of shear force, but so there is this variation along it, but it still means that you can have actually relatively high flow rates here without cell damage. Okay, and just on to so I've talked about all of that work has been with the hollow fiber bioreactors, but Moen is another PhD student in the group, and he's been working well, essentially on a reactor for to make whole cut cultivated meat, affectionately known as the CASP bioreactor, that has on his very early studies already is showing actually really a pretty decent, I would say, final cell density. So just to put this into context, a hollow fiber membrane is around 10 to the 8. Now this is a first iteration, pretty confident that we're going to get higher than this. And this is really good news, because if we can start to get structured scaffold and cell growth with such high densities, then this is going to make essentially making those constructs more efficient to produce. And Moen has shown that actually he's able to get a good cell distribution on seeding throughout that structure, which when you're thinking, when you've got essentially something that is relatively solid, or it's not because he's got channels to it, that historically in tissue engineering, seeding these types of structures has resulted in a necrotic core, and Moen has overcome this. So we get good seeding as we scale, so different sizes. So small, medium and large, which relates to small, medium and large, relatively speaking. There's a few dead cells here, but on the whole, we've got good viability and we've also got differentiation. So some early data, but very good signs that this slightly different bioreactor design could allow for that, that kind of whole cut meat structure. Okay, so that's everything I'm going to share with you today in terms of my slides. So just a few concluding comments to put all this together. So cellular agriculture as a field has the potential to simultaneously address a number of the UN SDGs, particularly zero hunger, climate action, good health and well-being. There's actually scope for many more than that because they are so interlinked. We're at a stage of development in this field, I think, that we can determine how we can collectively contribute to these goals. I would say we're all well aligned on climate action, but I think there's more we might want to think about around good health and well-being, and the kind of people aspects of what we're doing. So we're still early enough that we can do that. We've mentioned this a few times this evening, but we can build on the wealth of bioprocessing know-how, repurposing what there is already available and innovating further to realize cultivated meat production at scale. So all of those early slides I showed you about bioprocess design, that is, so that's kind of standard undergraduate level design. So we have all that available to us. There's a lot of ability. We can repurpose reactors that are already available on the market, and that's a really good place to start. But then we also can innovate further to realize and overcome the challenges for us to realize production at scale. We need to work together. I don't think I need to tell this audience that, but we need to work together across the sciences and engineering so that we can essentially diversify protein sources. And for scaled bioprocess design, we need to combine experimental studies, computational fluid dynamic studies, mathematical modeling, so we need to understand all of that to enable us to design a system to allow us to scale for cultivated meat production. So with that, I will just say thank you to everybody. I've mentioned a few of these people as we've gone through, in particular, Saeed, Scott and Moen, but there are many, many more people involved. So thank you to all of those. Thank you to GFI and the other funders of the work, without whom this progress would not be possible. And thank you all for listening.
Thank you so much, Marianne. That was a fantastic seminar, and I really appreciate you taking the time to share your knowledge and expertise with our community. I thought that was a fantastic presentation, and you covered so many topics while still, I think, making it engaging for a range of audiences. I certainly found the information really accessible, despite the fact that I do not have a bioprocess development background. And I especially just want to signal boost this last point you made around the idea that alternative proteins, one of our central value propositions is to build a more sustainable, healthier, more resilient protein supply. But we shouldn't take that for granted, that, you know, none of those things, whether we're talking about health or sustainability, are really built into the technology themselves. And we will have to work in concert with a range of stakeholders across the ecosystem, especially this early in the trajectory of our reimagination of this protein supply, to make sure that we do rise to meet that sustainability mandate. So thank you for that final call to action. I did want to make sure that folks saw a few different resources that I think might be useful to you all before we pivot over to Q&A. So the first is that GFI did recently release an LCA TEA for cultivated meat. So for those of you who want to dig a little bit more deeply into understanding what the projected environmental footprint of cultivated meat might be in 2030, say, and also what the pathway towards price parity might be. I recommend you checking out these newly released reports because they are, to date, the only LCA and TEA studies that incorporate data from players along the cultivated meat supply chain. So they paint a really helpful roadmap forward for the next decade or so. Marianne also noted the importance of an interdisciplinary approach to bioprocess development and scale up. So I also wanted to note the existence of the organization, the Cultivated Meat Modeling Consortium, which I've just dropped a chat link to in the chat there. So for those of you who do have a computational background and might want to contribute your skills to this line of investigation, feel free to reach out to the CMMC. And then lastly, for those of you who are interested in participating in a series of networking sessions, my colleague Renee Bell will be hosting a series of networking sessions for scientists in a month or so, and I'll drop the information for those networking sessions here in the chat as well. Okay, so with all of those little announcements taken care of, let's pivot over to Q&A. For those of you who saw Renee's message in the chat, you know that there's this feature where you can upvote certain questions. So feel free to go ahead and look through the range of questions that have been posed for Dr. Marianne Ellis and upvote the ones that you most want to hear the answers to. We have 22 questions so far. I'm sure we won't get to all of them today, but I'll try to consolidate a lot of the questions so that we get to the heart of what you all are curious about. All right, so one of the very, I think, common questions that I see throughout this Q&A section is about the selection of the hollow fiber bioreactor. So a lot of people seem to understand that hollow fiber bioreactors have been around for 4 or so decades, but we've been unable to scale them much beyond 2,000 liters or so. So how can we get anywhere near the economic feasibility at the scales needed for large-scale meat production? They seem to suggest that stirred tank reactors that are widely used across the biopharmaceutical space have already been maintained at 20,000 liter levels. So what is your thinking around selecting the hollow fiber bioreactor design as opposed to the stirred tank reactor design? And are there examples of successful scale up at the levels that we might want to see for cultivated meat production?
So there's I guess there's a number of ways to answer this, but I'm aware there's a lot of different questions, so I'll try to keep it short and happy to take follow-ups. So certainly stirred tanks would make sense right now because we have the technology and it is proven to scale. If you work out how much, essentially how many reactors you would need if you were to see a really significant replacement of current meat consumption with cultivated meat consumption, that's one hell of a lot of steel. And it may work in some instances. So, for example, there is a possibility that you can have a super kind of really fast proliferating cell line that is continuous processing, that you wouldn't need to have a lot of batches, or you could have not too many or not too large systems. But our current thinking is that actually it's not feasible at the scales we will have to be at, just in terms of the amount of material you'd need. And also if the concept as to how you build these factories, lots of factories, and where they're going to be, and say it comes down to land use as well. So if you remember that kind of whizzy picture I had under my vision and mission, one of my personal drivers is to be able to have cultivated meat production in places like refugee camps, favelas, in slums, in kind of high population areas where you need a small footprint, and you need a small plant, which means you need very space efficient units, reactors, or units, which for me drives us towards those high cell density systems. No, they haven't been manufactured on the very large scales, but it's not a comparable volume that you need to achieve in terms of size of reactor. It's a comparable number of cells that you need. So, you don't need to have the same size reactor for the same size number of cells with a stirred tank. You can actually have one that's about 200 times smaller for the same number of cells. So, you need to think about it more in the cell density as opposed to the overall volume.
Awesome. Thank you so much. Yeah, there's certainly room to explore different kinds of scale up versus scale out models. And I think we're so early on.
Yeah.
Yeah, awesome. Thank you so much for that set of considerations. There is a question here about how scalable you think the process of decellularizing grass or other plant materials for producing biodegradable or edible scaffolding is. Have you seen that process be scaled? And if not, what do you think the pathway forward is for scaling decellularization?
Yeah, it's a very, very good question. I think there is potential if you can recycle the materials that you're using within it. So it's this concept of net zero carbon. If we already have the chemicals or all the systems available, if we can kind of keep them out of the system and not having to keep have new renewables come into the system, then I think it is potentially scalable. Yes.
Awesome. And when you talk about scaling the bioprocess for cultivated meat production, what do you think is the optimal scale? Like, what do you really mean by large scale bioreactor?
Yeah. Again, this is again my perspective and kind of personal preference, and possibly more as an academic and not an industrialist, would be this idea of having something that can produce kind of more batches of sort of maybe a few, I don't know, I think in cell numbers, so maybe hundreds of kilograms, but locally. So you've kind of imagined that every, kind of almost microbrewery-esque in locations where people can pretty much walk to their local manufacturer. So that's kind of possibly a bit of a utopian view. Like I would look at small local farms in the same way that you kind of, I'm literally sat here looking across the valley at the sheep that are basically sold as meat in the local butcher. So that's kind of a personal preference, but I would also like to see, I guess, our technology or the technology being used for, I would say, for mass manufacturing, because ultimately that's what we need, is for this to be available to people to buy off the shelf. And so, Quorn have done it for one protein alternative. We're seeing, I think that's the scale that we would need to be working at, is tons, many tons per month to feed everybody.
Absolutely. Thank you so much. Yeah, there are some questions here about whether you have any insight into the successful engineering of semi-permeable hollow fibers for alternative or edible biomaterials.
So in theory, you can make fibers out of materials like alginate. People have done that, which is seaweed derived. You can get cellulose acetate. You probably want to eat that. So pure cellulose fibers. I know of some technologies that can form cellulose into other structures for other applications, but sort of pure cellulose membranes have potential. really what it comes down to is whether, first of all, so there's three parts of a system to make a hollow fiber or to make a membrane: the polymer itself, the solvent, so you need a solvent that can dissolve the polymer, and then you need a non-solvent, which then causes precipitation of the polymer out of the solvent, and that movement of the solvent out and the non-solvent in is how you get the porous structure. So if you can find the chemicals to do that, that will dissolve your basically your edible polymer, then yes, you can make fibers out of edible materials. Remembering when you're looking at the full broad life cycle analysis, and the product requirements, texture, taste, downstream processing, you need to balance them all up. But yeah, I'm actually all for edible scaffolds, and the cast bioreactor, we're aiming towards that being an edible scaffold, because that's a whole cut meat structure.
Awesome, thank you. There is a question here from Thomas about whether the ecosystem really exists kind of along the supply chain for bioreactor development. There's obviously a lot of work already being done on the bioreactors themselves, but do you think there's sufficient work on the enabling technologies like pumps and automated systems and other associated components? Or are the technologies that we're using, these enabling technologies, all inherited from biopharma? And is there a lot of room left for optimization for cultivated meat?
Yeah, very good question. I think given the I guess the vast array of equipment available, I think probably that is. I don't think for a long time we're necessarily going to need to develop that so much. There might be need for bespoke equipment, but I think we can go a very long way with what's already available. It's for me, it's more about getting the reactor design for that high density, like scalable high density system, and then it's a bit of a plug and play system with what's currently available. Having said that, be pretty happy if people were developing new positive displacement pumps that might be particularly effective for this type of system. But I suspect for a long time we'll be using what's currently out there. Happy to be proved wrong though.
Yeah, thank you. Something that I loved was that, one of the applications you talked about over the course of your presentation for cultivated meat technologies is the idea of refugee camps or other kind of land scarce applications. And there's a question here from an attendee, Kaja, who asks about how we can make cultivated meat technologies relevant to actually feeding the world when we talk about developing regions, for example, that certainly don't fall into this category of purchasing expensive high-end products, and cost and scale just mean very different things when we talk about the rest of the world beyond Western industrialized nations. So can you speak, and especially when we talk about global food security, 30 or 50 years down the road, most of the population growth that we anticipate seeing will be in developing regions in Asia and Africa. So how do you think we can overcome the bottlenecks to cost and scale that will allow us to make these technologies accessible to a global population?
Yeah, sure. So I think it's really important that the products are relevant to the people who are going to be eating them. So again, for example, so from the work that we've done with the various process designs with our master's students, we've got the Welsh plant, which is we'll be working more towards a pate, typically slightly higher end emulsified product. We've done work. We did an example, so the Turkana refugee camp example, that was actually acknowledging that that many of the people who are there locally, so not necessarily there in the refugee camps, but locally, are nomadic herders, nomadic goat herders. And one of their speciality foodstuffs is a mix of goat blood and goat milk. So that project actually had the idea of taking mesenchymal stem cells, selecting them, getting them from the blood, growing them up, and then providing a protein from that source. So it would fit in with the, with the processes that were already done. And so I think, kind of recognizing that the different food preferences and cultures is really key. And that actually is exactly why I was like, saying the point about that. As someone who's producing processing technology and others in the space, we need to understand what basically our customers, so people using the technology, want to make, because that has a knock-on effect. It definitely has a knock-on effect on the downstream processing, but it also therefore has a knock-on effect further up into the upstream processing.
Awesome, thank you. Yeah, definitely a lot of room to explore cultural applications for cultivated meat, and also to further reduce cost and scale and to build infrastructure in the relevant regions, certainly. I want to zoom in back to kind of down a few levels back to your research and the implications for your research. So there are a few questions here about what the process of harvesting tissue from the hollow fiber bioreactors might look like, and if you could speak to what the process is like.
Cool. Okay. So so currently, the team are using triple E to enzymatically digest the cells to remove them. So just as you would take them off tissue culture plastic, using that same approach, and then but then that adds in another component that you have to essentially remove and from the food stuff at the end. So I don't think that's ultimately going to be the way that we would go. You can potentially, in a hollow fiber system, use shear to remove the cells off the surface. So you can flow media around the outside to kind of to push them off. And then there is I guess the ultimate goal of having mature cells that come off the fibers and then are just collected and as they circulate. So that would be the ultimate goal.
Awesome, thank you. There's a question here from Steve about whether the CASP bioreactor uses scaffolding material or some other sort of materials for cells to attach to.
Yeah, it does. It does use a scaffold. I think I probably put the little star that I've been at pain of death. Can I give any further details, because we're looking to through the university to patent it. But yes, it is a scaffold, and so the approach that Marianne is taking to get pseudovascularization combined with the way that the scaffold is formed and essentially added into the reactor. Yeah, it's a scaffold-based system, but that, I mean, actually, it could be used for proliferation, but the goal is really to use it as a final stage, as the differentiation stage. But currently, edible scaffold.
Fantastic, thank you. And this flows nicely into the next question here, which is about whether you can explain the role of scaffolding in cell growth, and whether you think it ideally is removed from the end product, or if it becomes an integral structure of the final product, and how the biomaterial selection might impact considerations like texture and taste.
Mm-hmm. So muscle cells are naturally adherent cells, so they need to grow on something, they need to be attached to a scaffold. So at the moment, most cultivated meat work would be using a scaffold. There are some I guess groups, I think there's probably one company and another a research group who have, and probably more, working on this, developing a scaffold-free cell line. That would be the most ideal if all you want is I guess that the highest protein density that you can possibly get. So this is, when we're looking at having a culture adapted cell line. So we're looking back at CHO cells, Chinese hamster ovary cells, developed for the pharma industry. So and they took many years to develop. So I think ultimately we are looking for something that is scaffold free. Having said that, there are benefits of having a scaffold for the product. So one would be depending on the material, it can add fiber. And actually, you know, particularly in the West, we don't actually generally get enough fiber in our diet. So you could actually have it as formulating part of the fiber. You can also use scaffolds as carriers for other components. So either during the culture process, to release growth factors, for example. So you could have timed release or controlled release. You could also capture flavor molecules or other components in it for the product for downstream processing. So at the moment, I would say the scope, you know, I can see scope for both, but from a pure processing perspective, it would be nice if we could one day remove scaffolds.
Thank you so much. Those are some really excellent considerations. There are a few questions here about whether you can elaborate on this medium recycling loop. You did speak at one point about the importance of kind of changing media in order to control for nutrient depletion and other factors related to kind of cell death. So, and kind of the waste that's produced by our cells. But certainly, you know, changing media out will have repercussions for the cost of the final product, since media is supposed to be such a big proportion of the marginal cost. So there's a question about whether or not you can speak in more detail about what an optimal media recycling system might look like, such that we mitigate or balance the tensions between these two factors.
Yeah, sure. So our initial rationale, we first started looking at recycling actually was from a water reuse. So whereas we're, you know, I've mentioned net zero carbon, in terms of that water use as well, if we can recycle the water, particularly in places where there's not a lot of excess water, not an issue here in the UK most of the time, but that's, you know, we still get droughts here. And there's places around the world where you just don't have that much water. So if you can have a closed system on water and use water as a carrier, then that was probably our first point. But then, yes, of course, there is the recycling of any unused media components. So at the moment, media recipes are not such that everything gets used up equally. So you end up with some components that are left, and if they happen to be more expensive ones, you know, that's a whole other kind of conversation. Then you would definitely want to recycle them. But even the less expensive ones, when you're looking at scale, they still cost money. So if it may be cost effective and carbon effective to recycle, to collect those. And then you've got the third reason, which is the well, you've got the purge, or you've got the separation of the waste components out, so you can still have that recycle. So you would have to go through some kind of separation unit to remove probably lactate, possibly ammonia. You could just purge and replace, but those are the two that are most likely to need to be removed. I'm also aware that the sun has just set and it's gone really dark, and I'm nowhere near my light switch. So apologies if I'm kind of slowly going into the dark.
No worries. If you want to pause and grab a light switch.
Okay, I'll just go off video. It will take me 10 seconds.
Yes, take your time, please. I don't want you to be blinded by your computer screen. We have just a few minutes left here in our seminar, so we'll wrap up with one or two final questions and then give Dr. Marianne Ellis the opportunity to say some final words. She's back. That was so speedy. I didn't even have to buy us too much time there. So
apologies.
No worries. Fantastic. So just two final questions. One is about whether or not you took any biological measures of improved function after alignment. This question asker has noted that alignment probably doesn't necessarily equate to improved functionality. So they're not positive that having grooves is an inherently positive quality, and are curious whether you can speak to whether there was any function improvement.
Sure. Very fair question and a very short answer. No, we haven't. As I did say that this was some relatively new work, and so we haven't gone into function. So contractile function, for example. So I'm very sorry. It's a very quick answer, very short answer, but that is So it's important to do for that full understanding. And I alluded to the complex biology, and that would of course be part of it, would be the more functional aspect, and the and ultimately what we're interested in is the total protein content.
Absolutely. Thank you. So looks like there are definitely a lot of outstanding questions for other researchers to take on and for further extensions of your project. Thank you for helping identify some of those white spaces, especially because this audience is full of scientists and engineers that are looking for the right entry points into this field and the right questions that still remain for them to work on. So thank you for highlighting that. And this next question is very related to that. Our next and final question, there are several questions here about workforce development, and specifically coming from scientists and engineers who are really interested in entering this field. So the first part is from engineers like automation engineers or electrical or mechanical engineers without the biochemical, bioprocessing background. How do you think they can enter into the cultivated meat field and help close some of these white spaces? Do you have any insight into resources for jobs and internships and research positions focused on these white spaces?
Fantastic that people from all different disciplines are interested in joining us in this field and helping to advance it. If you have got the enthusiasm and the passion and the willingness to learn, then it actually doesn't matter what your background, expertise or studies have been so far. You can come in and you can learn. I would go as far as to say that if you had even a non-engineering background and you want to come into the engineering space, then you can. In our group we have a range of natural scientists and engineers who come in and all doing all using bioprocessing, developing bioprocessing. If you want to stay in your own field, I can't remember the types of engineers, but electrical engineers and others, then I think yes, there's plenty of scope. Actually, process instrumentation and control, biosensors, all of those aspects are going to, will allow for better process design. I think all the types of engineering activities that you will see come up in other industries, we are going to need in our industry. So come and join us and get in there early. In terms of resources, other than GFI's website, I would say also New Harvest's website, and specific universities, have a look and see who's active in the space. So you can do that, I guess, by actually just looking through who are all current grant holders. Have a look on the list, see who's attending here, and do some screenshot and do some Googling. Just contact individuals, but there are programs popping up around the world actually, and I think there's a growing number of those. Even if there's nothing specific, perhaps look at other related topic areas, which could be processing, it could be manufacturing, could be bioprocessing, could be pharmaceuticals, because actually those processing skills will be relevant to our field if you can't find or don't want to do a specific course.
Awesome. Thank you so much. I did drop links to a few key GFI resources that might be valuable to you. Our collaborative researcher directory can help shed light on others who are working in the field and in the relevant disciplines, and also who are open to collaboration. Our student resource guide can help shed light on some of these career pathways, and our Advancing Solutions for Alternative Proteins initiative highlights white space opportunities that are unique to certain disciplines. So would recommend that you check out all three of those resources. And the final part of this question that also pertains to workforce development as coming from a recent a master's in chemical engineer or chemistry graduate, excited about going into the cultivated meat sector, and is curious to hear your thoughts on how the PhD market in this field has been progressing in the UK over the last few years, and how you anticipate it might continue to progress.
It's exploding. It's fantastic. Yeah, there's so many more opportunities and so many more people working in this space in the UK. Reach out to the supervisors that you're interested in what they're doing, and there might be local sources of money. There's national sources of money, and and you've mentioned other opportunities as well through GFI and the like. So yeah, there's a lot of potential and opportunities in the UK.
Awesome. Well, thank you so much, Marianne, for joining us today. This was honestly such a joy, and I learned so much over the course of your presentation. Thank you for being so generous with your time and fielding these questions. Before we wrap up for today, do you have any final thoughts that you'd like to leave our scientific community with?
I think just work together to address the big questions, the big global questions, and we have the skills and the passion to make this world a better place through our contributions, in our case, to alternative proteins. So yeah, stay positive, and we will see all of this hard work coming to fruition soon, I'm sure.
Yes, we will. Love that last dose of informed optimism. Yeah, this was so fantastic. I had a bunch of fun moderating today's session, and want to thank our global community again from joining from almost every corner of the world. We will be joining, or we hope you will join us again next month for another GFI grantee presenting about their research on the plant-based meat side. So we will see you again soon. Thank you again to Dr. Marianne Ellis for a fantastic presentation and for driving forward such important research for our field. I hope you all have a great rest of your day, and we will talk again soon. Bye. Thank you.